Hi guys, welcome to my all-in-one biology video, and this is for EDEXCEL GCSE 9 to 1. I hope you find it super helpful. As with all my other all-in-one videos, I try and go through every single specification point. All my perfect answers flash up, and don't forget, if you want to buy my revision guide, which I have written, containing my perfect answers, you can get that on the website, which will flash up now. I hope you find this video super helpful. I hope your studies are going well. Don't forget to come follow me on Insta, Twitter and Facebook, because I add lots of extra exam tips, extra explanations and just genuine cool science stuff, particularly on Insta. So I really would recommend giving that a go, and I do go through past papers there too. So yeah, I hope you find this video helpful. Let's get started. Right, we're going to be talking about all things to do with plants, starting with that key plant process, photosynthesis. Remember, photosynthesis is carried out in the chloroplast of plant cells. They contain chlorophyll, which absorbs that sunlight and actually carries out the process of photosynthesis. And this is the method by which green plants make their own food. So let's start by looking at the word equation for photosynthesis. Remember that it's carbon dioxide plus water forms glucose and oxygen. This is why it's such a great process, because of the huge volume of oxygen photosynthesis releases. In terms of the balanced symbol equation, try and learn this off by heart, and remember that 6 is very important. So you've got 6 CO2 plus 6 H2O forms C6H12O6 plus 6 O2. Now, unfortunately, there are lots of different factors which can act to reduce or limit the rate of photosynthesis, and we call these limiting factors. You need to know the definition of a limiting factor. It's a factor which in a reaction is in the shortest supply, and a lack of this factor is the reason why a rate of reaction no longer increases. Now, in terms of photosynthesis, there are 3 limiting factors you need to know about. These are carbon dioxide, light, intensity and temperature. So any one of these may act to limit how much photosynthesis can take place, and we're going to talk about each of these in turn in a different situation. So let's think about early morning. We have a green plant and it's trying to photosynthesize in early morning, but what could be limiting how much photosynthesis takes place? Well, it's early morning, so it's quite cold, so obviously temperature is going to be a limiting factor here. Another limiting factor will most likely be light intensity, because it's not as light early morning as it is at midday. So despite carbon dioxide levels increasing, you will find that low light levels, low temperatures limit the rate of photosynthesis. The reason being is that in the morning, low temperatures obviously mean low kinetic energy, so all those enzymes involved in those chemical reactions involved in photosynthesis can't actually come together. They don't collide as frequently with their substrate molecules, and that's all because of little kinetic energy. If we take midday now, we know that the temperature will have increased, we know that the light levels will increase, so neither of these things will be limiting factors. The most likely limiting factor at midday will be carbon dioxide levels. And be sure that you can look at graphs on all of these factors and make sure you understand what is going on. So, we know photosynthesis is the method by which plants make food, and they make that in the form of glucose. So what do they do with that glucose? Well, remember that glucose contains carbon, hydrogen, oxygen. So it obviously contains components that can be used to make other biological molecules, and this includes fats and proteins. So that glucose is used to make fats and proteins. It's used to make storage compounds such as starch, which the plant can call upon in lean times, and it's also used to make the sugar cellulose, which is an important component of plant cell walls because it gives it its structural integrity. Now we're moving on to specialist plant cells. So we're going to start with the xylem. Remember, xylem transport water from the roots up the stem to the leaves where it's needed in photosynthesis. Xylem have very special structure. First of all, they're dead, and that means that they're hollow because they've lost their cell contents ages and ages ago. That hollowness means that obviously there's lots of space for water to be transported. They have special walls made out of lignin, and that lignin is a very strong substance which will help keep the xylem open. Now, coupled with xylem, you find that there are phloem. Remember, phloem transports sugars around the plant, so from the leaves where the sugar's made up and down the plant for storage. Now, you find that the ends of the phloem cells break down and form sieve plates, and those sieve plates allow the food to move easily in the water in which it's carried. Because phloem cells need lots of energy, they're coupled with companion cells. Now companion cells are full of mitochondria which provide the phloem with energy. Let's take a regular plant cell now. So we call this the palisade layer if we're looking at the structure of the leaf, or the palisade mesophyll. Obviously they contain lots of chloroplasts. Why? Because it's full of a pigment called chlorophyll which absorbs light in photosynthesis. So that's really, really key. You do find that these plant cells, or these palisade cells, are located near the top of the plant in order to capture as much sunlight energy as possible. And because, as with all plant cells, they have a large permanent vacuole, and that helps keep the cells rigid. Lastly, we're going to look at root hair cells. So if you're asked to draw a root hair cell, basically draw a plant cell but with a big elongated area, which is the root hair. And obviously don't include chloroplasts because it's a root, it's underground, there's no sun going to reach there, so it would look really stupid if you added chloroplasts. And just draw all the other structures as usual, your cell membrane, your cytoplasm, your cell wall, etc. And just point out that that root hair gives a very large surface area to allow all that water to be absorbed. Remember, it has a large permanent vacuole which helps speed up the water movement by osmosis. And again, they have lots of mitochondria. Why? Because they need lots of energy to actively transport minerals from the soil into the root hair. I'm just going to talk a little bit about transpiration right now. So remember, transpiration is the evaporation of water from the surface of the leaf, and that occurs through small pores called stoma or stomata. You will find that there are increased transpiration rates depending on certain conditions. The amount of water that leaves the leaf is based on the amount of water surrounding the leaf, because it's all to do with the diffusion gradient. So obviously water is moving from high concentration to low concentration. First of all, if it's dry, that means that there's very few water molecules outside the leaf because the air is dry, which means there's lots more water inside the plant, and therefore, based on what you know about diffusion, the movement of water will occur very quickly, so water will be leaving the stoma and you'll have high transpiration rates. Equally, if it's wet, if it's humid, there's a lot of moisture in the air, a lot of water molecules, so the difference between the amount of water inside and outside the leaf will be very little, so our gradient will be small and therefore transpiration won't be happening very much. Next, if it's windy, all these water molecules will have lots of energy and they'll be moving away from the leaf very quickly, and so you'll find that as the water diffuses out the leaf, it gets blown away quickly. And again, that means there's not very much water surrounding the leaf, so transpiration rates will be high when it's windy, and equally they'll be much lower when it's still air because the water particles, the water molecules won't be being blown away. Next up, if it's sunny, you'll find that transpiration rates are really high. Why is that? It's quite a complicated answer. If it's sunny, it means that lots of photosynthesis is taking place. What does photosynthesis require? It requires sunlight, but it also requires CO2. CO2 enters the leaf by the stoma. So in order to photosynthesize, the plant needs to open its stoma in order to allow that CO2 in. If you open the stoma, it means water will automatically start to leave. At night time, you find that the stoma tend to close because there's no sunlight, so there's no need to have it open and no need for CO2 to enter, so you will find that less transpiration happens then. At high temperatures, you find that transpiration rates are increased, and that's just because the kinetic energy of the water molecules is increased. And like the windiness thing, it means that they're just moving out of the stoma, out of the leaf, much faster. And if you're in doubt with this and you can't remember anything I've said or anything you've learned in your lessons, but you're in the exam, just think about the conditions which would help washing to dry, and you'll certainly pick up some marks. Now, the last thing I wanted to talk to you about was the potometer. The potometer is used to measure the rate of transpiration. Here's a really terrible diagram of a potometer. So what's happening in this diagram is that water is leaving the leaves by transpiration, and therefore in order to replace that water, you find that the stem draws in more water from the tube it's attached to, which is attached to a reservoir. And there's a little scale that you can use to measure how far the water's moved and how much water's been absorbed by the plant effectively. So you just read it off of the capillary tube, and that will tell you how much water has been absorbed. We assume that the rate of water uptake and the rate of transpiration are identical. Now, what sorts of things could you do to the leafy shoot in order to measure the rates of transpiration under different conditions? Well, you could put a bag over the plant in order to increase the humidity and hopefully see a decrease in the rates of transpiration and the rate of water uptake. You could use a hairdryer to emulate windy conditions, which would blow water away from the surface of the leaf. You could shine a light on the leaf. You could increase the temperature. So there's lots of things. However, in the exam, they like asking you about the setup of the equipment and any flaws and any ways you can fix them.
and how you can minimise error. So first of all, you want to avoid getting water on the leaves, so you might need to dry them if you accidentally splash them, and that's in order to prevent humid conditions being inadvertently given to your experiment when you're not actually interested in that. Second of all, you need to cut the shoot underwater, and that's to stop air getting inside the stem and preventing water movement, because you'll have an airlock. You also want to cut the shoot at an angle to make sure it has a really good fitting into the rubber tubing. It's important to use Vaseline to seal the joints in order to stop air getting in places where it ought not to be. Finally, you use the capillary tube to magnify the amount of water uptake so you can actually see what's going on.
So we're going to start by looking at two vessels, two tissues you need to know lots about. That is the xylem and the phloem. Make sure you can label these both inside the root at cross-section and inside the stem. Note that in the root the X matches with the xylem, which is why the X in the middle of the root is xylem. That's a nice way of remembering. Whereas the circles around that X are the phloem. Inside the stem it's slightly different. The outer layer of tissue is the phloem, the inner layer is the xylem. Try and remember this because the phloem transports sugar, and aphids are little insects which bite into that stem in order to steal some food, so they're biting in so that they can reach that phloem and take the food, so the phloem is on the outside of the stem. So looking more closely at the roles, as I've already told you, phloem transports glucose, and it transports it from the leaves where it's made in photosynthesis to other parts of the plant, so to growing regions such as flowers and the tops of stems, and to storage regions such as the roots, and that's where it's stored as starch. Carrying on with the plant theme, we're now going to look at the structure of a leaf and how it is adapted for photosynthesis. Let's make some generic comments at the beginning by stating that a leaf has a large surface area, which is obvious, so that it can absorb more light. It's thin, so the gases don't have to diffuse too far. Looking very closely now at the structure of the leaf, you need to be able to label all those layers and discuss which each of them do, and you need to be very specific here. So we're going to start with the waxy cuticle. The waxy cuticle is there to prevent transpiration, and remember that transpiration is the loss of water from the leaf. So a nice thick waxy cuticle prevents excess water loss. Next up we have the upper epidermis. Remember, this is transparent and it allows light to enter the leaf. You don't need to say anything more than that. The layer beneath this is the palisade mesophyll. Mesophyll is just a fancy way of saying tissue. The palisade mesophyll is what your generic plant cell looks like. So it's your rectangle block. It's crammed full of chloroplasts, so it contains lots of chloroplasts for photosynthesis, and this is where photosynthesis takes place. Under this you have the spongy mesophyll. The important thing to note here is the presence of plenty of air spaces which allow gases such as carbon dioxide and oxygen to diffuse. You'll also find the vein here, and the vein contains the xylem and the phloem. The xylem brings water into the leaf, the phloem transports sugar away from the leaf. Then we have the lower epidermis. Not a lot to say here. The next layer is the most important layer, and that contains the guard cells and the stomata. Now the guard cells control whether the stomata are open or closed, and the stomata allow carbon dioxide into the leaf and oxygen and water to leave the leaf. Let's quickly look at the role of diffusion in gas exchange in plants. So remember that carbon dioxide is needed by plants in photosynthesis, so it will diffuse into the leaf through the stomata. Oxygen is released by photosynthesis, so that will diffuse out of the leaf, also by diffusion. However, don't forget that plants are living organisms, so that they respire, which is a bit confusing, which means they need some oxygen, obviously, in order to carry out respiration. So if you look at it over the course of the day, you will find that during the night, because the plant can't photosynthesize, because obviously there's no light, it will just be respiring, which means that more oxygen will enter the leaf than will exit the leaf. During the day, photosynthesis tends to occur at a faster rate than respiration, so in this case more oxygen will leave the leaf compared with diffusing into the leaf. So now we need to look at how plants adapt themselves to living in extreme environmental conditions. So that could be extreme heat, extreme coldness, extreme dryness. So we'll start by looking at the deciduous plants. And deciduous is simply a posh way of saying plants which lose their leaves in the winter. So what are they adapting to? Well, they're adapting to cold temperatures and frozen soils, so they can't really obtain water that well. So how do they do this? Well, I've already told you what a deciduous plant is. It's one which loses its leaves. And why do they do that? In order to conserve water. Let's look at a second type of plant, which is the conifers. Now, they have to withstand some pretty extraordinary extreme conditions, such as cold temperatures, high winds, and dry high temperatures. So obviously this will be different conifers in different parts of the world. So how do the conifers which exist in environments which are extremely cold survive? Well, they have needle-shaped leaves, and this helps them to prevent water loss. If you think about it, somewhere it's really cold, it's likely to snow a lot, and plants really don't want snow settling on their leaves. So having needle-shaped leaves will help with this. How do they cope in high winds? Again, they have needle-shaped leaves. Because they have a smaller surface area, it means they catch the wind less, so they're less likely to get blown over. Next up, if they're found in dry, high temperatures, they have their stomata. So remember, stomata is where water leaves the leaf. These stomata are hidden in pits, and what that does, it helps to trap water vapour around the leaf, giving an impression of high humidity, which means that transpiration occurs more slowly. This is very strongly linked with the transpiration topic, so make sure you check that out if you're struggling with this one. And lastly, I want to look at cacti. Now, these are obviously found in dry, hot places. They have lots of adaptations, such as their leaves become spine, and that reduces water loss. Many of them have tiny hairs, which help to trap water vapour around the plant. The stomata are also found in pits, and that's for the same reason as I gave here. It's to increase the humidity around the leaf, which decreases transpiration. So just write that here again. And lastly, they have a thick cuticle, and stem stores water. And again, that's to prevent water loss.
We're going to look at tropisms. First of all, what is a stimulus? And that is a change in the environment. So what sort of stimuluses do plants need to respond to? Clearly they need to respond to the amount of light, and they actually also respond to gravity. So we call a plant's response to light a phototropism. A plant's response to gravity is known as geotropism. So how do organs of a plant react to various tropisms? So let's take phototropism first of all. Clearly, a stem shows positive phototropism. Why? Because it grows towards the light. Roots show negative phototropism because they grow away from the light. Let's look at gravity now. Roots obviously show a positive geotropic response because they grow down in the direction of gravity, whereas stems show a negative geotropism because they grow away from gravity. Looking more closely now at how these changes are brought about to the stem and the roots, we need to understand the role of auxins. So what are auxins? Well, they are plant hormones. Let's now explain how a plant stem may bend towards the light, towards the sun, because you do see particularly sunflowers bend towards the sun. That's because the auxins concentrate on the side furthest from the light source. This causes cell elongation on that side, so the cells get longer, and therefore the plant stem bends towards the light. They do like to show you various experiments, and an important part of this will be the coleoptile, which are these really boring little seedlings. Now note that they are cereal seedlings, and they are simple plants used to investigate tropisms. So do be aware of how they respond if you chop their tops off, if you put a mica sheet in between the top of the stem and the bottom of the stem,
if you use agar jelly, you do need to know different responses. Lastly, the role of a clinostat. This is another boring piece of equipment. So a clinostat is a device used to remove any stimulus. So for example, it negates the effect of gravity or negate the effect of sunlight. Now looking at adrenaline, remember that is the fight or flight hormone. It's released in bucket loads when we're under conditions of stress. And it's good to imagine a really angry cat when you're thinking about the effect that adrenaline has on your body. Because if you imagine the cat, then it will actually help you remember every single thing that happens to you when you have adrenaline coursing through your body. So this cat's hairs have stood on end, and that's because it wants to appear scarier to its opponent. Its pupils have dilated, and that's to allow more light into its eyes so it can see more clearly. Its heart rate, not that you can see this, but adrenaline has caused its heart rate and breathing rate to increase. Why? The heart rate increases to deliver blood faster around your body. Your breathing rate increases to allow more oxygen into your body for respiration. Blood is diverted from your digestive system to your active muscles in your arms and your legs, and that's so you can either fight or run away if needs be. So yeah, try and remember the cat, at least for the hair standing up on end and the pupils dilating.
Female reproductive system, you have 2 ovaries, and then it joins to the fallopian tubes which link with the uterus, and then that links at the bottom to the vagina via kind of like a trapdoor called a cervix. The cervix is usually closed, and then obviously it opens when you have a baby. You need to know about the menstrual cycle. Being a cycle, it means it starts and finishes, and it happens every single month. For around 28 days is an average length of cycle, but that can obviously vary from woman to woman. Right, so we're going to start by naming some hormones because that's always a good place to start, and I'm going to start with FSH. So FSH is follicle stimulating hormone. Now these follicles are basically the eggs inside the ovaries, and it's kind of like before they're fully mature. And so by definition, follicle stimulating hormone kind of means it's the hormone which causes the follicles, the eggs, to mature. So if you're asked in the exam the role of the FSH, I would say that it causes the eggs in the ovary to mature. FSH's second job is to stimulate the ovary to produce estrogen, and that's another very important hormone. But first of all, I'm going to talk about luteinizing hormone, LH. Now, luteinizing hormone is a very important hormone because what its role is to cause ovulation, and ovulation is the exact point at which the egg is released from the ovary. So if they ask you what ovulation is, you say the release of an egg from the ovary. What is the role of LH? To cause ovulation. So at that point, the egg pops out and it will travel down the fallopian tube. Next up, we need to talk about estrogen. As I said before, estrogen is produced by the ovary. Now, estrogen is really important because it causes the uterus lining to build up, and it's important that that uterus lining becomes very thick in order to support the egg, which, if it gets fertilised, will implant itself. Progesterone is essential because it maintains the wall of the uterus, and without progesterone, what would happen is the uterus lining would flake away, and that would be a period. So it's really important that at all stages during pregnancy that progesterone levels remain really high. It's worth noting that estrogen also inhibits FSH production. That's really important. Why? Because if you're building up the uterus lining in order for a zygote to implant, the last thing you want is more eggs in the ovary maturing.
We now need to look at methods of birth control. So this is a way of avoiding getting pregnant. They can be split broadly into 4 categories, which is natural, chemical, barrier, and surgical methods. And they all vary in terms of how effective they are. Starting with natural methods, the first obvious thing you can do is abstain. So this is called abstinence, and this is basically when you don't have sex, because clearly if you don't have sex, you can't get pregnant. You can also monitor your body temperature and also your cervical mucus levels, because these vary over the menstrual cycle, and at times you'll be more likely to get pregnant and less likely to get pregnant. Although it's worth noticing here that these aren't the most reliable means of controlling whether you get pregnant or not, because obviously body temperatures tend to fluctuate regardless of menstrual cycle, and the same with cervical mucus levels. Obviously abstinence, if you don't have sex, you can't get pregnant. So moving on to chemical methods, you've got IUDs, and this stands for intrauterine device. So this is a device placed within the uterus which prevents the zygote from implanting in the wall of the uterus. And it could be a foreign object, and often they're called coils, which is, I think it's made out of metal, which is a little metal object placed inside the uterus. The fact that there's a foreign object means that that zygote won't implant. Similarly, intrauterine systems, they work in a very similar way. Next up, we have the contraceptive pill, the injection, and the implant, and these are all really similar. So the pill is something that most people have to take every single day. It contains a combination of progesterone and estrogen, and by maintaining those hormone levels, it means you can't get pregnant. An injection is just something which is given once every 3 months, which contains the hormones in a specified amount. And an implant is a small device placed under the skin in the upper arm, which releases hormones slowly over time, and all these methods will prevent you getting pregnant. The barrier methods are probably historically the ones which have been used the longest. So we're looking here at condoms, and as the name suggests, it's a barrier to the sperm, so it stops the sperm reaching the egg. So condom is obviously a plastic sheath worn over the penis that stops the sperm being ejaculated into the vagina. A femidom works in a similar way to a condom, but is found inside the woman's vagina. And lastly, a diaphragm is a small plastic cup that sits over the cervix, so the entrance of the uterus, and again acts as a barrier, preventing sperm reaching the egg. And then you can go very hardcore with surgical methods. So this could include a vasectomy, which is when the sperm ducts in a man are cut and tied. So obviously there's no longer a tube allowing the sperm to pass from the testes to the urethra, and therefore they can't possibly leave the body. And female sterilisation, which works in a similar way but will involve the female's oviducts rather than the sperm duct. To go into a bit more detail about the hormones found within the contraceptive pill, sometimes you find that there are progesterone-only pills, and all these do is they prevent the zygote implanting. Whereas progesterone and estrogen combined pills prevent ovulation, so they actually stop the egg being released in the first place, and therefore tend to be a better way of preventing pregnancy. Now we're looking at the opposite, so looking at becoming pregnant, and if we're not going to do normal sex, we can look at alternative methods such as artificial insemination, and this is all to do with sperm donors. So a man may decide to donate his sperm, and that gets stored in what's known as a sperm bank. I think the sperm are actually frozen, and then when the time comes, when a woman is close to ovulating, so when she's at her most fertile, then that sperm can be inserted into her vagina and she can become pregnant in the normal way. And remember that sperm can live for up to 3 to 4 days within the woman. IVF now that's quite famous because it's a method used to help couples who are struggling to conceive, and that's actually known as in vitro fertilization. And what happens here is the ovum or the egg is fertilized outside the body. Tends to happen inside a laboratory, and then once that egg has been fertilized, it's placed within the uterus of the female. And that's been an amazing way of helping couples who previously couldn't have a baby conceive.
Let's look at the role of the skin now. So first of all, the skin acts as a barrier, and that actually helps prevent pathogen entry, because we often have viruses and bacteria landing on us, but because the skin is such an amazing intact organ, it does actually prevent those microorganisms entering our body. It's also waterproof, which is why you don't swell up when you go swimming. It forms a very tough outer layer just generally to prevent you from physical harm if you knock into something. It's a sense organ for pressure, touch and pain. And lastly, it controls our heat loss. How? By either sweating or the hair stand up in order to trap some insulating air close to our bodies. Let's now look at what happens when we're too hot and too cold. So we're looking further at the homeostasis topic, which remember is all about controlling a steady internal environment within our bodies. So let's start by looking at when we are too hot. Well, first of all, an uncomfortable thing happens, which is that we sweat. The reason being that when sweat evaporates, it acts to cool the body. Vasodilation occurs, and that's the arterioles in our face. They dilate, they become wider. What that means is that the blood flows closer to our skin, and therefore heat can be radiated more easily. And also when we're too hot, the hairs lie down on our body, on our arms, and that's just so that less insulating air is trapped close to our skin, because remember, air is a good insulator, it keeps you warm. So by making our hairs lower, it means we're kept less warm. So if we're too cold, clearly our hairs will stand up on end, so that more insulating air is trapped close to our skin, and that actually acts to keep us warm. We shiver, the reason being that muscle contraction releases heat energy. And lastly, the opposite of vasodilation occurs, vasoconstriction. The arterioles in our face constrict, they narrow, bringing blood away from the surface of our skin, so less heat is radiated.
Let's look at how blood sugar levels are controlled, and we need to know lots of hormones and the effect that they have on blood sugar. We're going to take two scenarios. So first of all, after a meal, clearly your blood glucose levels will increase because you've just eaten. It's important that we reduce those blood glucose levels. So the pancreas, remember which organ does this? So it's the pancreas, releases the hormone insulin. And that insulin travels to the liver and causes it to convert soluble glucose into an insoluble compound known as glycogen. And as you can see, blood glucose levels will therefore have decreased. After exercise, or when you haven't eaten for a period of time, clearly your blood sugar levels will have decreased. So glucose levels have decreased. The pancreas again is involved, and it releases a hormone, this time called glucagon. And please notice the spelling of glucagon and glycogen. They're incredibly similar sounding words, and it's so essential you spell them correctly, because I know there'll be a point in the mark scheme being like reject if they've spelt it wrong. So make sure you take pains to learn that spelling correctly. This time the glucagon travels to the liver, and it causes the insoluble glycogen to be converted back into soluble glucose, which then gets released into the blood, and subsequently you see an increase in blood glucose levels, which is all good. Now, famously, a disease when you don't control your blood sugar levels appropriately is called diabetes, and there's type 1 and type 2. Type 1 tends to be the one you get as a child, and we call it early onset for this reason, and it is inherited, so you get it through your genes, so you inherit it from your parents. And here you'll have to do insulin injections because effectively your pancreas isn't producing enough insulin. In terms of symptoms, so how you might know that you have diabetes, first of all, you'll get excessive thirst, so you'll want to drink all the time. As a consequence of that, you'll produce lots of urine because you've taken on so much water and liquid. You get increased hunger, sweet smelling breath, and the biggest giveaway is if the doctor tests your urine and there's glucose in the urine, that can be an indicator of diabetes, because really we know that the glucose should have been reabsorbed in the proximal convoluted tubule of the kidney, so glucose in the urine would indicate that there's something wrong.
Moving on to excretion, so both in plants and humans. Obviously, when we're talking about humans, we'll be really looking at the kidney. Let's start by looking at the definition of excretion. It's the removal of waste products of metabolism from the body. Looking at excretion in humans, what sort of substances are excreted? First of all, sweat from the skin, urea from the kidneys, carbon dioxide from the lungs, Do note that faeces are not excreted, they are ingested, and I already touched on this in the nutrition part of this video. So remember, with the kidney we have the nephron. This is when you take the kidney and you zoom in and you see that there's a Bowman's capsule followed by the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and then lastly the collecting duct. So let's have a look first of all at what happens at the Bowman's capsule. That is a process of ultrafiltration. Ultrafiltration is when small molecules are filtered out of the blood into the Bowman's capsule. Such molecules include urea, sodium and chloride ions, glucose and water, and remember that these are forced out under pressure. So how does the structure of the Bowman's capsule and the blood vessel flowing into it, the glomerulus, actually aid this ultrafiltration? The reason is because the blood vessel entering the glomerulus is wider than the one leaving. This creates a huge amount of pressure which actually forces those small molecules out of the blood into the Bowman's capsule. Which molecules don't enter the Bowman's capsule? Well, that's proteins. Why? Because they're too large to pass through the basement membrane. Now we've entered the proximal convoluted tubule, we need to know what happens there. The process of selective reabsorption occurs here. Now this is when all glucose, some ions, and none of the urea is removed from the proximal convoluted tubule back into the blood. This is occurring against the concentration gradient, so it requires energy. Now we're moving on to the collecting duct, because we now need to look at water reabsorption, and this is all to do with osmoregulation. Now osmoregulation is the control of water content in the blood, and obviously sometimes we drink lots, so we need to wee a lot. Sometimes we drink very little, so our wee will be very, very concentrated and small in volume. And now we're going to look at the mechanisms involved in controlling the amount of urine we produce. So let's start when we've not had very much to drink, so overnight for example, on a hot day. Clearly our bloodstream will contain little water. That passes in the blood to your kidney, specifically the collecting duct, and it basically tells the collecting duct to be more permeable to water. So it adds lots of channels into the collecting duct, which enables lots of water to leave the collecting duct and be reabsorbed back into the blood. This means that there's far less water left over to create urine. So in this situation, urine is very yellow, it's concentrated and low in volume. Let's look at the opposite effect. So when we have had lots and lots to drink, it makes the walls of the collecting duct less permeable to water, meaning that more water stays inside the collecting duct and that less is available to be reabsorbed. This means that there is more water flowing from the collecting duct to the bladder, and therefore resulting urine is high in volume, not very concentrated and very pale in colour. Just to touch on the name of some key vessels, the vessel linking the collecting duct to the bladder is known as the ureter. The vessel taking urine out of the body, so from the bladder, is known as the urethra. And if they ask you in men what two substances does the urethra transport out of the body, that would be both sperm and urine. Definitely not water. Some people in the past have written water. That is really silly. That is not the answer. Looking at the general structure of a kidney, you can see the outermost layer is known as the cortex, the middle layer is known as the medulla, and then the ureter passes out, and then we've obviously got the renal artery and the renal veins supplying and removing blood. Moving on to the breathing system, so we are looking at our lungs and all the vessels relating to that. So we're going to start with our mouth. It leads down to our windpipe, which we're going to have to call the trachea. The trachea branches to form two bronchi. Further branching occurs, which is the bronchioles, and that ends in lots of air sacs, which we call alveoli, and they're surrounded by a network of blood capillaries. Now, if you actually think about it, it is very much like a tree, because the trachea is represented by the trunk. The big branches are the bronchi, smaller branches are the bronchioles, the alveoli are similar to the leaves. So do bear that in mind. Now we need to talk about how those bronchi and bronchioles are kept clean. That's due to the presence of two types of cell, the goblet cell and the ciliated cell. The goblet cell first of all secretes mucus. That mucus is good because it traps bacteria, pathogens, and it stops them from entering your lungs. This is when the cilia come in, because remember they have hair-like projections which waft, and they waft that mucus, which is covered in bacteria, up to your mouth where it can be swallowed and destroyed by your stomach acid. Now, what is ventilation? Well, that's simply taking air into and out of your lungs. Looking more closely at the alveoli, because this is where gas exchange takes place, where oxygen moves into the blood, carbon dioxide leaves. We need to look at the adaptations of alveoli for gas exchange. So clearly they have a very large surface area. They are thin, which gives you a short diffusion distance for that oxygen and carbon dioxide to move across. They are moist, which helps the gases to dissolve. And I can't think of anything else. So that is the adaptations of alveoli done. We're going to start by looking at the components of blood. That will be red blood cells, white blood cells, plasma and platelets. Now remember, plasmid is that liquid which actually acts as a suspension. It carries these various cells around the body. What sorts of things are transported in plasma? These are going to be our products from digestion, so things like glucose, amino acids, so soluble products. There'll also be hormones being transported. There'll be urea from the liver, which needs to be taken to the kidneys so it can be excreted, and carbon dioxide. Platelets, these are small fragments of cell. They clot the blood at the site of a wound, so they're very important in forming scabs. They convert soluble fibrinogen into insoluble fibrin, and that forms the meshwork which seals the wound. Looking more closely at red blood cells, we need to look at the structure of red blood cells so we can see how they're so well adapted for their function. They have a biconcave dish shape. This means they're donut shaped, and what this does is it maximises the surface area to volume ratio, ensuring that they can transport as much oxygen as possible. The absence of a nucleus also means that there's more room for oxygen. And do mention that they contain a pigment called hemoglobin, and that hemoglobin binds to oxygen, forming oxyhemoglobin. Next topic, white blood cells, and they are the phagocytes and the lymphocytes. So starting with the phagocytes, remember that they engulf or ingest pathogens by enclosing them inside a vacuole, and then digestive enzymes are secreted which destroy the pathogen. The second type of white blood cell is the lymphocyte. The lymphocyte is far more complicated, and it works by recognising the antigen on the pathogen. It secretes lots of antibodies which destroy that specific pathogen, and in this way the pathogen is destroyed. Now, it has various modes of action which help to increase the pathogen destruction. First of all, it labels the pathogen, making it easier for the phagocyte to recognise it and therefore engulf it. It neutralises any toxins produced by the pathogen, and it also causes the bacterial cell to burst open on occasion. Lastly, it makes the pathogens stick together. With that answer relating to the lymphocytes, notice that I use lots of keywords, antigens, antibodies, for example. Try and include as many keywords as possible. Just shove them in your answer, because if you look at mark schemes, they'll be underlined as being worth a mark each, so it's worth writing them in anyway. Don't just keep repeating yourself. You need to insert lots and lots of keywords here. Looking now at the structure of arteries, veins and capillaries. So let's start with the arteries. They have a narrow lumen. Remember, the lumen is the hole in the artery. It's like the hole in a straw. So it's narrow. This clearly means that blood is going to be forced through at a high pressure. Because it's at a high pressure, it means that the walls of the arteries need to be very thick to withstand this pressure. Be nice and detailed here and state that they have thick muscle and elastic fibre walls. Looking at veins now, in veins blood travels at a much lower pressure. This is because veins have a much wider lumen. The walls therefore need to be much thinner, so they have thin muscle and elastic fibre walls. They contain valves, and these valves prevent the backflow of blood, because that blood sometimes travels so slowly it's liable to start backing up. We don't want that to happen, so the valves force it to move on in the right direction. Capillaries now. Capillaries are the tiny vessels that supply all our cells with oxygen. They are 1 cell thick, and this enables a very short diffusion distance, and they have an extremely narrow lumen. Let's now look at transport in animals. So now we're going to talk about the circulatory system, so we need to look at the heart. I've already talked a little bit about multicellular versus unicellular organisms. The reason why organisms such as ourselves, humans, require a circulatory system is because our surface area to volume ratio is too small and diffusion is too slow. So we need a circulatory system which actually acts to transport oxygen around our bodies. So that's the reason why we have a circulatory system. The heart forms the epicenter of our circulatory system. It is the pump which delivers oxygen around our bodies, and you've got to know detailed information about how this actually happens. So we're going to divide the heart into 4. Do remember that we switch over the left and right sides when we're looking at a diagram. Why? Because we're picking up the heart and pushing it into our bodies. So it is the opposite way around if you actually think about it. So the 4 chambers are the left atrium, right atrium. They form the top 2 chambers. Left ventricle and right ventricle form the bottom 2 chambers. Now do remember that pulmonary means relating to the lungs. So if you have used the word pulmonary, it means that blood must be flowing either to or from the lungs. So we're going to start by picking up oxygen in the lungs. It's going to be delivered to the heart, to the left atrium, via the pulmonary vein. Remember that veins bring blood to the heart, arteries take it away. So the pulmonary vein delivers oxygenated blood to the left atrium. The left atrium contracts, forcing blood into the left ventricle. Do you remember that the valves open here, and they are the bicuspid valves? They open to allow blood to flow from the atria to the ventricles. The left ventricle contracts, forcing blood into an artery. This is the aorta. It is the main artery delivering oxygenated blood around the body. So that blood goes and delivers oxygen. The oxygen is removed and used by respiring cells. Clearly the blood will now be deoxygenated, and it needs to return to the heart so it can be pumped on further to the lungs. So it's going to return to the heart via a vein. The vein is the vena cava, and it's going to take blood into the right atrium. The right atrium contracts, forcing blood through the tricuspid valves into the right ventricles. The right ventricle contracts, forcing blood into an artery. This artery is clearly going to be flowing to the lungs so the blood can be oxygenated, and this is why it is the pulmonary artery. So that's our overview of blood flow around the body. Let's notice a couple of things about the heart. Firstly, that the walls of the ventricles are thicker than the walls of the atria. Why? Because they need to pump at a much higher pressure to deliver the blood much further. After all, they're delivering blood to both the lungs and the rest of the body. All the atria are doing is pumping to deliver the blood slightly lower, a couple of centimetres lower into the ventricles. Why is the wall of the left ventricle thicker than that of the right? Again, it's a distance thing. Blood from the left ventricle is going all around the body. Blood from the right ventricle is simply returning to the lungs. Your heart is here, your lungs are here. It's not too far. Why do we call this system a double circulatory system? Well, that's because the blood passes twice into the heart for every once it travels around the body. Simpler organisms such as fish have a single loop, so the blood just keeps passing from the gills to the heart around the body, round and round and round. They're not as efficient at oxygenating their bodies as we are. You've got to know quite a lot about the various vessels that travel around the body. Just remember that arteries carry blood away from the heart, and do remember that pulmonary means relating to the lungs, and that hepatic means relating to the liver, renal means relating to the kidneys, so renal failure, kidney failure, hepatitis, disease of the liver. So these words, if you do know what they mean, it really helps, because then when we're looking at what's the name of the vessel supplying the liver, well, it's hepatic. It's coming from the heart, which means it's an artery, so it's the hepatic artery. What is the name of the blood vessel entering the kidneys? Well, it's coming from the heart, so it's an artery. It's going to the kidneys, so it's renal, so it's the renal artery. The vessels leaving the organs, well, these have obviously got to return to the heart to become oxygenated, so they're going to be veins. So the vessel leaving the liver will be the hepatic vein. Just note the hepatic portal vein, that's just the name of the vessel which shunts blood from the digestive system to the liver, and that's the only weird one you need to know about. Let's look at how a heart rate increase is brought about during exercise, for example. So clearly when you exercise, you're going to produce more carbon dioxide. Why? Because your muscles are respiring more. So that carbon dioxide flows in the blood, and it is detected by both the aorta and the carotid artery. This sends impulses or messages to the brain, particularly the medulla part of the brain, and specifically the accelerator nerve, Now that accelerator nerve causes an increase in heart rate so that more oxygen can be delivered to your muscles and so that more carbon dioxide can be removed. The next topic we need to tackle is respiration, both aerobic and anaerobic respiration. Remember, it's the process carried out in mitochondria which releases energy. So what is this energy used for? Well, it's used in cell division, it's used to build up large molecules from small molecules, it's used in active transport, and it's used to contract our muscles. Let's look at the equation for aerobic respiration now. So you need to take oxygen into the body, you need to add that to glucose, an arrow, and then what's produced is carbon dioxide and water, and energy is released. Again, you need the balanced symbol equation here, and it's again all the sixes. I hope you've noticed that photosynthesis and respiration are the same equation, just reversed. So in terms of the balanced symbol equation, you're looking at C6H12O6 plus 6O2, arrow 6CO2 plus 6H2O plus energy in a square bracket, if you so wish. Now, aerobic respiration involves the use of oxygen, and that's the type of respiration that we carry out ordinarily. Now, sometimes we have to carry out anaerobic respiration, which tends to be when we've exercised and when we can't take enough oxygen into our bodies, and this involves the incomplete breakdown of glucose, and we find that lactate is produced instead. Now, lactate is pretty poisonous. It's what gives our muscles cramp, and we need to remove that lactate, so we have to take in more oxygen, which we call the oxygen debt, in order to break down that lactate to ensure the glucose is completely broken down. Now, there are two places where anaerobic respiration takes place that you need to know about. So I've already mentioned one, which is in your muscles. So I've given you all that information about the oxygen debt. It's when you can't take enough oxygen into your body, and that's because you've been doing something pretty strenuous like sprinting. You also need to know about anaerobic respiration in yeast. Remember that yeast is a type of fungus, and yeast anaerobically respires and it breaks down glucose into ethanol plus carbon dioxide, and these are very useful industrial processes because remember the carbon dioxide is used to help bread rise, and that the ethanol is used in beer making. Ecology now, not my favourite topic, mainly because it's full of disgusting definitions which all seem to be very similar and all sound the same. So let's start by looking at the definition of environment. That is the total non-biological components of an ecosystem. So we're looking at the soil and the water, for example. The habitat is the place where a specific organism lives. Now, population, be very specific with your keywords here. This is all the organisms belonging to a particular species which you find within an ecosystem. What is the community? This is the population of all species found within a particular ecosystem. Now, what is a producer? Because remember, producers start all food chains and food webs. This is just a plant which photosynthesizes to produce its own food. A consumer is an animal which eats other animals or plants. What is a decomposer? It's an organism which decays dead material and helps to recycle nutrients. Define a parasite. This is an organism which lives within another organism, causing harm to that organism and feeding off of them. What is a predator? It's an animal which kills and eats another animal. Gosh, these definitions do keep coming. What is biodiversity? That's the variety of plants and animals found within an ecosystem. What are biotic factors? These are living factors. So these are living factors which affect organisms' lives. So it could be other animals competing for food, competing for nesting sites, bringing disease and pathogens to other organisms. Abiotic factors are non-living factors which affect organisms such as soil pH, temperature of water, carbon dioxide availability, number of daylight hours, etc. So now we've looked at all the definitions, we now need to look at how we're going to sample an ecosystem. So say we've got a big field and we want to know about this variety of species living there. How are we going to do that? And we are going to use a quadrat. So remember, that is a big metal frame which you're going to place randomly on the field, and you're going to take a sample of the organisms you can find within it. Make sure you can draw a quadrat. It's not a complicated diagram, just draw a nice metal grid. And be prepared to state how you would use it. So first of all, you need to place it randomly using a random number generator, because what you do, remember, is you get a field, you effectively mark it out with imaginary squares, and then you use your random number generator to work out where to place that quadrat. Don't say throw it, because that's really biased. It means that it will ping off to the left or the right. So don't say throw the quadrat. You're going to write down all the species you find within the quadrat, and then you're simply going to repeat and place it in many other places around the field, so you've got a really good feel of the place. Looking more closely now at pyramids of numbers and pyramids of biomass. So remember, a pyramid of numbers simply shows you the number of each organism at each trophic level. So trophic level is just the stage in a food chain. So for example, a pyramid of numbers could start with grass. The grass could be eaten by rabbits, so that will be the next tier, and lastly you'll have foxes. But we don't like using pyramids of numbers because often they end up looking a really strange shape and not being pyramidal at all, and that can be due to the producer only being one organism, such as a tree. That will be very small in comparison to the number of sparrows living on it, which is why you end up with very funny shaped pyramids. So we use pyramids of biomass because that actually shows the mass of living material available, and therefore the oak tree, for example, will appear much larger, and therefore the pyramid will be the correct shape. I'm just going to talk you through a couple of food web questions just so you know how to answer those.
The diagram shows a food web from a habitat. Use information in the food web to complete the table. The first one has been done for you. This has been drawn quite horribly, so rather than looking at where the layers are, you're going to need to count the arrows and make sure you remember things about what a producer is and what a primary consumer is. So starting with the number of organisms, which they've done for us, which is annoying because that's the easy question. Just count how many different organisms there are 8, so we do agree with them. The number of different types of plant. You're looking for the producers here. So what here is a plant? Well, you can see from the picture, even if you don't know what a cattail is, that it's a cattail and marsh grass. So the answer here is 2. The number of animals, so these are the animals which form anything from the primary, secondary, tertiary, consumer level. So the animals therefore are grasshopper, cricket, shrew, frog, snake and hawk. And if you count those up, you'll get 6. Number of primary consumers, so remember they appear straight after the producers. So we've got an arrow leading to the cricket and the grasshopper, which is why the answer here is 2. Number of food chains, God, this is more difficult. So food chain 1 will be marsh grass, grasshopper, shrew, hawk. Food chain 2 is the same but includes the snake before the hawk, so that will be the second one. Food chain 3 is the cattail, cricket, shrew, hawk. Food chain 4 will be the same but include the snake. And then food chain 5 will include cattail, cricket, frog, snake and hawk. So that was pretty tricky, but the answer here is 5. The plants in this food web make the food for some of the animals to eat. Give the name used to describe these plants. I've already mentioned that a lot. That is a producer. The hawk catches and eats its prey. Give the name used to describe the hawk in this food web. So I told you earlier in my definitions that something that catches and eats something is a predator. A pesticide can be used to kill the grasshoppers in this habitat. Describe the effect that killing grasshoppers would have on the number of shrews. So we need to have a look. Oh right, so the grasshoppers are eaten by the shrews. So clearly if we reduce the number of grasshoppers, the number of shrews will reduce because there is less food for them. Describe the effect that killing grasshoppers would have on the number of marsh grass plants. So if we have fewer grasshoppers, we can see that they won't be eating the marsh grass as much, which means that the marsh grass population will increase.
Why is so little energy passed from one organism to another? So for example, from the producer to the primary consumer. So let's take grass, the producer and a rabbit as an example. Remember, the rabbit is the primary consumer. The issue here is that only part of the grass is digestible. Much of it passes out of the rabbit as feces. Some of the grass isn't eaten, so the rabbit won't even eat the roots. I don't know if that's true, but it can be a reason. The rabbit moves, it keeps itself warm, it respires, it digests, I've already mentioned this, it poos. So these are all ways in which energy is lost within a food chain. In fact, 90% is lost at each stage of the food chain. If they ask you where all that energy originates from, remember that is the sun. Let's now look at human impact on the environment, and we're going to start with eutrophication, which is an effect brought about when farmers use too much fertiliser on their land and when sewage, this is disgusting, washes into rivers and streams and lakes. So do remember, fertilisers and sewage contain a lot of nitrates. So what happens is the plants use those nitrates to build proteins and they grow extremely quickly. Because they've grown so quickly, they end up dying. The reason for this is due to lack of light. Basically, they block all the light for each other and they die because they can't photosynthesize. My notes are gone. The death of the plants obviously provides lots of food for decomposers and microorganisms. So they grow hugely in number because they're feeding on this dead material. Because they're aerobic respirers, they use up all the oxygen in the watercourses, and this means there's no oxygen available for aquatic animals and they die, and that's how leaching of nutrients, of fertilisers and sewage can end up with death of all aquatic animals within watercourses.
The good things and the bad things about fish farming, and also how you would do it. So what you would do firstly is you would feed the fish on a high protein diet. That's always worth a mark. You would need to control any disease, so you may need to add antibiotics, and the reason why disease will be more prevalent is just because the fish is so close together. The water needs to be very carefully controlled in terms of the temperature, oxygen availability and the build-up of waste, because that could lead to disease. There should be pesticides added to the water in order to kill any parasites, and you need to make sure that no predators can actually reach the fish and kill them and eat them that way. In terms of disadvantages, well, of course you've got issues with antibiotics being fed to the fish, and actually what that may do is it will end up in the food chain, and there's a lot of question marks over whether humans want to be consuming these things. The other thing is that the fish have to be fed little pellets which are made of other fish, and it's actually estimated that more wild fish get killed as a result of trying to feed the farmed fish compared with if we didn't have any farmed fish, which is obviously ridiculous.
Let's do the carbon cycle now. I do like this topic. If you just learn it as a list of steps, it's far easier than learning it as the whole cycle, unless you're a very artistic kind of pictorial person, because I really struggle in that way, but I find learning this list of steps works well every time. So we're looking at how carbon is cycled in our atmosphere and within living organisms. So the place I like to always start is carbon dioxide in the air. So what happens to that carbon dioxide in the air? Well, it gets absorbed by green plants in photosynthesis, and it is used to make glucose. Those green plants then respire because they're living organisms, and that releases carbon dioxide back into the atmosphere. So here's the first step of CO2 moving in, CO2 moving out. The plants are eaten by animals, and so that carbon that was part of the plants becomes part of the animal body, and then the animal respires again, releasing carbon dioxide into the atmosphere. Lastly, plants and animals inevitably die, and then this is where decomposers come in. They break down that dead material and they respire, again releasing carbon dioxide. So we can see carbon dioxide went in the first place with green plants photosynthesizing, and then it left via respiration by plants, animals and microorganisms. Do notice that combustion, which is burning of fuels and things, also releases carbon dioxide into the air. Now the nitrogen cycle, again start in one place and allow your answer to feed in from there. So I like to start with nitrates in the soil. So remember, those nitrates get absorbed by active transport into the root hair cells of the plants. Nitrates are important in the plant manufacture of proteins. So the nitrogen in the air becomes locked up in proteins within the plant. The plant is eaten by animals, and therefore the nitrogen moves from the plant into the animal. And then both the plants and animals die and microorganisms become important again. So the ones you need to know about are nitrifying bacteria, and they convert the ammonium within the dead matter from nitrites to nitrates. So nitrates have been returned to the soil, nitrogen has been returned to the soil. Do notice there are some really annoying bacteria called denitrifying bacteria, and what they do is they take nitrates in the soil and convert it back to nitrogen in the air. So they're not very popular with farmers because after all, the farmer wants lots of nitrates in the soil. They don't want nitrogen gas being released into the air because there's plenty of nitrogen in the air. So that's why denitrifying bacteria are particularly frustrating. The last thing to notice is nitrogen fixing bacteria, which we find on root nodules, so small bumps on the roots of leguminous plants. Legumes are a family of plants which include peas, beans and clover. So they're super good to plant because they provide a lot of nutrients to the soil. The reason being that the root nodules containing nitrogen fixing bacteria, they convert nitrogen gas in the air to nitrates. So effectively they make the plant its own supply of fertilizers. So that's really great because by planting clover or peas or beans, you're basically fertilizing the soil. So say the farmer regularly plants wheat as their cash crop, they might choose to crop rotate and plant clover every 4 years just to add a nice input of nitrates to the soil. Next topic, sewage treatment. Lovely. So first of all, what are the steps involved in water purification? You can write loads on this, but I want to make sure you're learning as least as possible for the most marks possible. So simply, all you have to do is filter to remove solids, and secondly, you need to add chlorine to kill bacteria, and we know that that is a super essential step. If they ask you what desalination is, it means the removal of salt from the water. Oh, another really bizarre bit, which is describing the process of reverse osmosis in water purification. Remember that membranes are needed to separate the dissolved salts from the salty water, and secondly, remember that high temperatures are needed, so it's a very high energy cost process. Faces! Lyra! So to this end, what is the disadvantage of using distillation to purify water? Kitten! Oh my goodness, twice in one video! So cute! So kitten, what is the disadvantage of using distillation to purify water? Well, it needs lots and lots of energy, doesn't it? Which means it's a very expensive process. Kitten. Kitten, are you really muddy? No, you're quite... Okay, bye. So if they ask you about sewage treatment, the 3 main steps, it's very similar to water purification. So you need to filter to remove solids, you need to add bacteria to actually feed on the sewage, and you need to carry out sedimentation, which is actually the process which makes sewage sludge. It's hard to say sewage. I think I'm saying it wrong, more and more wrong the more I say it. So how does using fertilizers increase crop yield? Well, the addition of fertilizers to the soil replaces leached or lost nitrates and mineral ions from the soil, because remember, fertilizers are very rich in nitrogen, nitrates, and those nitrates are used by plants to build proteins. What is a pesticide? Remember, it is a chemical which kills pests, so anything which feeds off plants will be counted as a pest. Killing pests obviously reduces their damage to the crop, and it also helps to increase crop yield. Issues though, there's lots of issues with using pesticides. Firstly, that they can be very expensive. They're persistent, which means it takes a while for them to decompose. So once you apply them to your soil, you've got to be aware that they may hang around for many, many years. And the problem here is that they can often kill animals which aren't even pests, which is really, really bad, because these are innocent animals getting killed by the pesticides because the pesticide does not discriminate correctly. So what happens here is it kills other animals. Some of these animals get eaten by larger animals, so we're talking about food chains here. And this is called bioaccumulation, where the pesticides become stored in these animals, and then as this pesticide works itself up the food chain, we call this biomagnification. And the famous case study of this is DDT, which was used to eradicate malaria and typhoid in the Second World War, and there are still areas of the world where DDT is killing huge amounts of flora and fauna, and that just means animals and plants. Right, I hope you found my video helpful, guys. These are so difficult to make, but I know you guys really like to just sit and watch the whole thing in one. So please give me a like this video if you found it helpful. It is a good incentive for me to continue my work, and don't forget to sub. I'll be back soon with another video. Bye!